Alright, today we're going to be talking about herpes virus vectors. This is in particular for gene therapy and other applications, probably some oncolytic virotherapy, which is using viruses to be a treatment for cancers. Alright, so the first thing is that herpes virus is a double-stranded DNA virus, and it replicates in the nucleus, which makes sense because it needs the nuclear machinery to help replicate its DNA. There you go. That's as opposed to RNA viruses, which just use the cytoplasm because they bring their own polymerase and replication materials. This virion is a little bit different. It has an envelope, but then it has tegument, which is this layer between the envelope and the capsid, which encloses the genome. So that E is the envelope, T is the tegument, the C stands for the capsid, and then the DNA is within that capsid, which you actually can't see in that picture up on the right. Genomes are about 120 to 240 kilobase pairs, and they encode 60 to 120 proteins. So this is bringing a lot of genetic material for something as small as a virus, and a lot of proteins. Where do I know that's a lot? Because something like influenza, which I am currently working with, is somewhere around like 10 to 12 genes and encodes about 8 proteins. So yeah, it's quite a bit. And this is widespread in vertebrates. All humans are infected with just some herpes viruses. Cold sores, when you get them, that's a herpes virus. They have a latent cycle, so they'll first infect you and you might see the disease as like a sore or something, and then they will just disappear and it'll appear to you as the host that they're cleared, but really they're just lying latent inside a cell and not having an active infection cycle, and they have various genes that help them stay hidden basically at the time. Alright, so herpes viridae, that's the name of the family, hence herpes virus. They are mammalian, avian, and reptile, and each group is quite a bit different. They're called by alpha, beta, and gamma. Fish, amphibians, and bivalve, bivalve meaning oyster, also have herpes viruses based on morphology, but they're not closely related to herpes viridae. And so we care in particular more about probably alpha and beta than gamma. Yeah, herpes simplex virus is within the alpha family, and cytomegalovirus is within the beta. Alright, so there are 8 human herpes viruses, and these are the standard ones. There's probably more that either are poorly studied or just don't have a huge impact on human health. HSV1, this is one we'll be referencing quite a bit. Type 1, it's in the alpha family, about 150 kilobase pairs, and it's the oral ocular lesion. In severe cases, which I just learned about recently, they can become encephalitic, meaning the swelling of the brain, or infection of the brain. HSV2 is the other main one we're going to be talking about. It's the same, except this is the one that is the sexually transmitted disease or infection. VSV is, or VZV, not VSV, wow, is the chickenpox virus, which if you get it or have it in your body as an older person, you can develop shingles because your immune system is going down. And so the latent infection becomes an active infection, but the infection is different because your body is different at that time in your life. Epstein-Barr virus, we'll talk a little bit about Epstein-Barr virus, possibly in a different video. This causes infectious mono, and it can be involved with tumors, not always. HCMV, human cytomegalovirus, this is congenital infection, and this is an infection when somebody receives a transplant from somebody else. Is a common time for it to happen. Then 6 is is in the beta subfamily, and it has a slightly larger genome size. Importantly, human cytomegalovirus, you'll notice, is the upper limit of the genome size, 230. This is at about 160, so it's a little bit bigger than the 1 and 2, but not that much. Rosella, yeah, we talked about. HHV7 is in the beta subfamily, and this sometimes causes Rosella. It's not important to our discussion. And 8 is Herpes sarcoma virus. It's in the gamma subfamily, like Epstein-Barr, and this is a little bit smaller than most of the others. But this is a known oncogenic virus, so this can cause cancer when it infects you. And it's used in a lot of research. Alright, the herpes virus replication cycle for HSV1. First you have the attachment to the extracellular matrix. Then in 2, you see it finding its actual receptor. In 3, the envelope fuses and releases the virion and capsid particles as well as tegument into the cytoplasm. I'm not going to go through 5A. Oh no, I will. 5A, it's using the microtubule system to move. 5B is showing that those tegument or capsid proteins are getting imported as well. Those have to be tegument and other probably non-structural proteins. The capsid moves along the microtubule, and then the capsid will attach to an entrance to the nucleus and deposit the virion's genome into the nucleus, which will then have first have mRNA made off of it that is exported to the cytoplasm and creates proteins. And these are separated into immediate, early, and late. Then there will be those proteins will get, some of them will come back into the nucleus, and then they'll start activating new transcripts, and that will be the early proteins, and then the late will be replication-based proteins. Eventually you'll get packaging of the genome, and that will cause that will be exported from the nucleus into the ER. There will be a first membrane, and then it will be exported from the ER to the Golgi, and with no membrane, and then once it reaches the Golgi, it'll get a new membrane, and then be exported, and then I think it just keeps the Golgi membrane. Then HSV infections in humans. This is what I'm talking about the possibility of getting a encephalitic reaction. So on the bottom right, it's a rarer thing to get serious disease from HSV infections, but it's possible. HSV means herpes simplex virus, or herpes virus, and that means that I will just be using that acronym because it speeds up this presentation by quite a bit. So in the normal maintenance cycle, you get a primary infection across a mucosal surface, so like in your mouth, down your throat, in your nose, whatever, or through an urogenital tract infection. But then you'll get your latent infection in sensory autonomic ganglia, so that's basically neurons in your face. But if they get far enough in, they can, if you follow the bottom arrow, you can then get into the brain, and then the brain's immune system is not well equipped to handle it. The initial infection can lead to widespread viremia, which can lead to your liver and spleen, and depending on how bad the infection is, that can cause serious damage. So we're just going to go through some background, some things about vectors, and the types. So, vectors. HSV is very easy to grow in culture and easy to engineer. That's because of that huge storage capacity. Its genome is really large. That's what I just said. Its potential to insert large amount of foreign DNA, and Amplicon technology increases that potential, and we'll talk about Amplicons later. HSV1 is neurotropic, so you can target neurons, and a lot of people, that's what they try and do, is they're targeting neuron-based diseases or neuron-based cancers. HSV1 undergoes latent infection in neurons, and so you can express therapeutic genes in the neuron without a subsequent infection because it knows how to hide there latently, and theoretically you could just turn on the latent genes to be on forever so that you'll never turn on a new primary infection after the initial one. HSV1 does not integrate into the host chromosome, so you won't cause insertional mutagenesis, which is the primary problem with something like a retroviral vector. So this large genome that you can put a lot of foreign DNA into, about 152 kb genome, approximately 80 genes, so we're talking about HSV1 probably. So that's about what? It's roughly 20 kb per gene. That's just what I'm telling as a rule of thumb for remembering what HSV is. However, in actuality, you can see those little different markers. Some genes are huge while others are barely visible on the map. So it's a way to remember it's not how it is in actuality. So about 40 of the genes are core genes that you need to have, and these are conserved among the herpes viruses. And they're things like the replication machinery, processing and packaging, because putting something inside of the capsid is not a type of machinery you can steal from the cell, because they don't put things inside of capsids like this. Integument definitely isn't found there. But the other 40 genes are dispensable, at least in tissue culture, which is where most of these are being made. Tissue culture is where you take something like a cancer cell and then you grow it essentially in a dish, and if it's from a human, then it will act fairly similar to a human cell, because obviously you can't do growing all of these things up in humans. Amplicons are a way to package foreign DNA inside HSV1. So these are bacterial plasmids that have the HSV1 ori and PAC signals. Ori refers to an origin of replication. PAC signal is the packaging signal. Very clever naming. If amplicons are present in an HSV1 infected cell, they'll be replicated as concoctamers and packaged into the virus particle. So you can get these plasmids inside of virus particles, and that's what you want. And so what you would probably, I'm guessing that they'll do, because this is a common theme in making these vectors, is you would infect with an HSV1 that has no packaging signal in its genome, but it has all of the other requirements of a virus, whereas this plasmid you would transfect into those cells, and then it will have the PAC signal in it. So these little amplicon vectors will get packaged into the new virus that's being created rather than the wild-type virus genome. HSV ori and packaging sequences mediate viral genome replication and packaging, which is what we just talked about. And so in HSV1 DNA replication, there are cis-active signals, the RIL and RIS, that will work together. And the origin is recognized by EOL9, the origin binding protein, so that's probably one of the genes you can't get rid of out of an HSV1. That helps recruit 6 other viral proteins involved in DNA replication. And so it's the basic set that you'd expect: a polymerase, single-stranded binding protein, a helicase primase complex, and then a processivity factor, which is probably acting in a way similar to the mammalian pCNA, which essentially forms a ring around the polymerase and other molecules to keep them on top of the DNA, otherwise they could fall off during the course of replication and not finish it. A model for HSV1 DNA replication: early synthesis is theta replication, late synthesis is rolling circle replication, generating head-tail concoctamers. So theta replication is basically opening the origin, and then rolling circle replication is basically a continuous cycle that's on the same circle. So, rolling circle. So you get this circle, so you get your initial nick right here, and then you'll start to get replication, but you'll start moving this old DNA off, and then you can get synthesis on here. So this circle that you have stays there, and then it's just continually making new genomes. So here's your first copy, here's your second copy, and you'll get concoctamers out of that you need to cut. HSV origin and packaging mediate viral replication and packaging. So that ORI and PAC sequence will tell if they're supposed to do rolling circle or theta replication. Packaging of the HSV1 genome depends on cis-acting PAC signals necessary for recognition of genome and its cleavage. Cis-acting, I realize, is a weird term. It means that it acts on that the PAC gene inside one genome acts only on that genome. So usually the way people have cis is humans have 46 chromosomes. If something acts only on the paternal chromosome 1 and at the top, and then it affects something at the bottom, that's still cis-acting because you're in the same chromosome. But if something on chromosome 1 would affect chromosome 14, that would be something named trans. When you're talking with viruses, it's on one copy of the genome versus another copy of the genome. Packaging is necessary, or this PAC signal is necessary for recognition of the genome, and then its cleavage. So, like I said, you get multiple copies of the genome from this rolling circle, and you need to cut them, and the PAC signal tells you how to do that. The packaging machinery feeds in 152 kb capsid, and then it cleaves it, requires direct repeats of PACs. So there's probably a couple of PAC genes right next to each other, like this, which are fairly close. And that's analogous to how T4 packs its DNA, which is a bacteriophage that's very famously studied. So step 1, you have this portal recognize the PAC signals. Then step 2, it starts pushing it in. Step 3, it's all in. And then step 4, it cleaves based on having the multiple PAC signals. Amplicon vectors allow plasmid sequences to be packed into HSV virions. 1 second, I have to Amplicon vectors allow plasmid sequences to be packaged into the HSV virions. So this means that you can put something that's not normally in the HSV virion. So you can put in your therapeutic gene of interest or suicide gene of interest or what have you. First A picture, so you have the HSV1 ORI sequence, then the red arrow probably represents a gene that you want to put in, and HSV1 PAC signals packaging symbol, and then green can be a different gene. And so in B you see a couple of different sizes, a 5 kb, a 15 kb, and 150 kb. And this is because a HSV virion, the capsid is your limiter, and so normally it's limited to 150 kilobase pairs, which is usually just one genome, but you can split that 150 into whatever you want. So if you want to have a bunch of 5 kilobase pair segments, then you could fit 30 of those inside of it. If you wanted to put 15, you could put 10, or if you had one, just really large one, you could put that in. That's 150. These usually have a marker gene like GFP. GFP is green fluorescent protein. It's taken from a bioluminescent bacteria, but it allows you to image if you're getting expression in a cell, because you'll know if you can see green under a fluorescent microscope that you indeed are getting expression of your plasmid. I'm just checking my battery quick. Okay, so we can continue this, no problem. HSV vectors, the features and advantages: easy to grow in culture. You get the Amplicon technology, which we just talked about. So you can get a huge amount of foreign DNA inside of them compared to something small like influenza, which can't be used as a vector, but small things like measles. HSV1 is neurotropic, and so you can target neurons, and that's probably what this is mainly used for. So the first two things are very generally useful. If it's easy to grow in culture and it's easy to engineer different types, so you can try a lot of different things, and the Amplicon technology allows you to put in a lot of those things. So neurotropic means you can target neurons, but the flip side of that is that it's probably hard to target non-neuronal cells. It undergoes the latent infection in neurons, so you can express these genes in the neurons. And insertional immunogenesis, again, is a good thing to avoid if you can. Because it doesn't matter if you fixed a disease if you just cause cancer, because people will not be happy with you. HSV naturally invades the peripheral nervous system, so things in your peripheral nervous system are like your sensory receptors for touch or your pain receptors, as well as things with your autonomic reflexes. So anything that controls a muscle, like a skeletal muscle, so on your arms and your feet, wherever, those are all going to be peripheral neurons. The exception is the gray areas on your face, and these can invade those neurons, like your trigeminal. Viral DNA in latently infected neurons are not integrated, endless, as in they exist as a circle, and they are nucleosomal, meaning that there are when they're latent, they hide out in the nucleus, but as soon as there's a lytic infection where the virus is breaking out, you won't find HSV DNA in that nucleosomal structure. It'll be more free-ranging than that. And the copy number is 10 to 1,000 per infected nucleus, which is fairly high compared to a seminar I just saw where they were marking for HSV and they only found about 8 genomes per nucleus. HSV1, genome transcription in latently infected neurons. So that LAT gene, the LAT, that is specifically a latency gene. None of the lytic genes are expressed. It's just this one 2 kilobase transcript known as latency associated transcript, or LAT, LAT. LAT is a non-coding RNA that's not required for latency, but does regulate the efficiency of latency. So this gene is being expressed as an RNA molecule, but instead of being exported to the cytoplasm and making some sort of protein, it's staying as an RNA that acts probably in a some sort of base pairing manner that allows for efficient latency. So if you don't have it, you can still get latency, but it won't be as efficient, and you'll probably get more lytic cycles, and therefore immune reaction. So the goal of a normal virus is to infect with a primary infection and then not come out until it can somehow either sense that there's a struggle with the immune system that's weakened the immune system, or some type of signal from the cell that the area around it is damaged or something like that. That means that for the virus to survive, it needs to escape to a new location. Why use HSV as a vector? We've talked about these, and we just talked about undergoing latent infection in neurons. So, we are moving on to types of vectors, probably. Yep, types of vectors. You can either get the Amplicon plasmid, or you can use the viral genome. Use recombination to put in a gene of interest, and you can do that to attenuate the virus and use it that way. Attenuating is passing it in cell lines, so it's not as infectious in humans or disease-causing in humans. Or you can just create a totally defective vector which doesn't have the replication genes. Attenuated HSV-1 vectors, their major application is in oncolysis. So this is using them to essentially attack cancer cells. Wild-type virus is known to replicate in both cycling and non-cycling cells. Cycling means that they're moving through the cell cycle and replicating. And it's specifically replication that defines something as cycling. So the strategy here is to delete those viral genes that you need for the viral replication in non-cycling cells, but they'll still be present as provided by the cancerous tissue. So some of the ENs utilized are thymidine kinase, or ribonucleotide reductase, the virion host shutoff gene UL41, and ICP34.5. And there can be effective antiviral drugs like acyclovir and ganciclovir that treat adverse effects in patients. But you need to have thymidine kinase functional for the drugs to work. So you usually you leave the thymidine kinase gene intact, just because you need them for in case something would go wrong with this application. And so thymidine kinase you kind of want to leave there, like ribonucleotide reductase, which is involved in replication. If you eliminate that, then all DNA-based things that are replicating require that, so it will be provided by the cell. Virion host shutoff. I think that just means that you won't stop replication anymore. And ICP34.5 is an important gene that's used a lot for this, but I am unsure of its actual ability. I assume it's related to ribonucleotide reductase in that it's something that's essential for replication that the host cell provides an equivalent for. So the current state is they have a great safety record, and even if something goes wrong, you still have acyclovir as a treatment. However, the efficiency of replication starts to become an issue with these, because as you engineer mutations or completely remove most of the viral genome, you reduce its efficiency, so you'll get some cells expressing it, but not a whole lot. They may be more effective when combined with chemotherapy, though that is still under debate. And the direct intratumor injection is the preferred route of administration, which limits the delivery to accessible sites. So for something that's already metastasized, HSV hasn't worked. And it's probably because most of us have circulating antibodies to HSV, as well as it's neurotropic. So it's not necessarily going to reach every kind of cancer. And so current clinical trials, TVEC is the big one, which Amgen is buying out from BioVex because the phase 3 just completed. Melanoma trial wasn't as effective as they'd hoped, but it's still got a pretty good thing. So it's ICP34.5 negative, meaning they took that gene out, and it has GM-CSF, which tells the bone marrow in the body to start producing certain immune cells. Seprevir, I am not as familiar with, but it's another ICP34.5 negative, and this is for lung tumors, and I don't think the results of that have come out yet. G207, same deletion. This is for brain, and this is specifically was chosen because HSV is neurotropic, and those results are still not out at the time I'm recording this. HF10 is an HSV, looks like an attenuated strain. And so this is, they're hoping to get some kind of systemic administration, I think, or they're doing 4 different trials. And that one, that will be interesting to know the results of, because if it's simply an attenuated virus, that means we can do without these deletions and just grow up this wild type or variant strain. So a particular promising candidate is that first TVEC, where they took out ICP 34.5, and this ICP 47 gene in particular is now HM, or not H, is GM-CSF. Not sure what this H refers to. The actual molecule they're releasing is GM-CSF. Never mind, I'm wrong. This H refers to human because you want to use the human GM-CSF because the immune system really differs between different species. So you couldn't use like chicken GM-CSF. I'm not sure if chickens have a similar enough immune system to even have a homolog. This oncolytic virus in a healthy cell will fail to replicate because there's no ICP 34.5 or equivalent, whereas in a cancer cell, which is represented by this orange business, ICP 34 will be replaced, and that will allow the virion to replicate, and then you'll get lysis. Presumably you remove away of the latent genes like LAT or LAT. TVEC clinical trials, there's a phase 2 melanoma trial that's fairly effective, 26% response rate that shows complete recovery. There's probably more responding to the therapy, but not as effectively, which is always the problem with chemotherapeutics and treating cancers, because all cancers are completely different. Even within the same tumor, you'll have so much mutation that something that might be really effective against tumor on the outer shell might not affect something on the inside of the tumor at all. So this phase 3 trial is OPTI-M, which is the one that just completed and caused Amgen to buy out TVEC. So you have an injectable, unresectable stage 3 B4 melanoma, and then you give up to 4 mils of this. So this is the plan for these. So you can put 2 to 1, so 2 times the number of people that are getting the virus treatment, and 1/3 of the people are getting a placebo or the current treatment. So usually with a virus, you have the current chemotherapeutic approach plus the virus. So these people who are not getting the virus, it's not that they're not getting treated, it's just they're not getting the virus. And then this is them adding in GM-CSF or checking for GM-CSF. So you can look at the disease, look at prior to systemic treatment, the site of the disease at the first recurrence, and the presence of liver metastases. So this is probably in mouse. This is probably not the clinical trial I misspoke. Or if it is the clinical trial, it's just the design. It's not actually telling us the results. Alright, TVEC can increase durable response rate. That's what DRR stands for. And it's defined as a complete or partial tumor response lasting at least 6 months and starting within 12 months of treatment. And so you start to see every group that got TVEC was more responsive than those who just received administration of GM-CSF. So this was the clinical trial. TVEC shows the trend to increase survival, and that's, you can start to see here, you're getting more survival, but you'll note that it's still a ways away from being 100% effective. It's, let's see, 30% of the people were living at whatever this in study months is, 45 study months. So it's an increase of about 10% or maybe 15%, but it's still not going all the way back up to a healthy individual. So this is not a significant difference. That's what trend means. It's probably a matter of numbers. No, they've got quite a few. 141 versus 295. There's probably a real difference between these two that's not coming through these numbers, but it's very clear you need a more effective therapy than this, and that's probably Amgen's next step now that they own it. So we just talked about one of the types of vectors. This would be probably an attenuated vector. So this is a defective vector. Its major application is in actual gene therapy. So you have something like dystrophin that you want to that's involved in Duchenne's muscular dystrophy, and you just need one viable copy into a host cell for whatever disease state there is to be reversed. So the strategy is to delete one or more of the essential genes of the virus so that it can't replicate, as we talked about before. But you can propagate this in complementing cells that express that native gene. And the more genes you take away, the less likely you'll get some type of reversion event where the virus becomes infectious again, but you'll decrease the efficiency of the virus. So you're always balancing efficiency versus effectiveness, or efficiency versus growing conditions. Non-replicating virus can be engineered to express the therapeutic gene in the nervous system. And this is, again, being restricted to that nervous system. It's useful because not a lot of viruses can make it through the nervous system, at least as far as we know. But it also means that if you don't want to target the nervous system, this might not be the vector for you. So there's a bunch of diseases which have some type of genetic component, such as epilepsy, MS, Alzheimer's, Parkinson's, and a bunch of different lysosomal storage disorders that are related to neurological function that are lesser known. Important to note is that of those diseases, a lot of them we don't know the root cause, so putting in this gene is effectively going to be a palliative effect until we know more. I'm not a neurobiologist, so we may be closer than I think we are to knowing these. So you put in these replication defective vectors. The important thing about that is you'll only get into whatever primary cells you infect. And so in those first neurons, in those sensory ganglia, you'll get things, you can put in things like BDNF, which is a brain-derived neurotropic factor that may help stabilize against epilepsy, MS. IL-4 is an immune system signaling. MS, some people believe it's, or not some people, it is an immune-regulated disorder, and it's probably, this IL-4 recruits a certain type of T cell and causes it to be made, so it's probably trying to force that T cell fate, as opposed to the incorrect response that leads to MS. Alzheimer's, I'm not as familiar with. shRNA refers to small hairpin RNA, which may be something that turns off a gene, and then neprilysin, I don't know what that is. Doesn't matter what those are, but before I move on, just note that you can use this for a number of different things, putting in some therapeutic transgenes, but all of these are preclinical except for chronic pain, which is just at phase 1, which is the safety and dosage escalation trial. So these are all right on the verge of us really understanding what happens when you put them in a human and if they actually have an effect on these disorders. NB2 and P2 is an HSV1 vector for treating chronic pain. So this is the chronic pain. It's creating pre-pro enkephalin, at least in that particular system. So ICP4 and ICP27 are put in, and enkephalin gene is located in the ICP4 locus, replacing it. So basically, you're cutting out a gene to put a new gene in, and it happens to be the replication gene that you're getting rid of. So this is a nice little system. So it's logical. Take out the replication gene. Where you're going to put in your new gene, probably where that gene was, and then you can use the promoter from that gene, transcriptional control if you would like. Otherwise, you have to mess with the virus more. This is to get you a poly A tail, intron, probably some splice variants that are possible. Phase 1 clinical trial of an HSV vector for treating chronic pain. So this is, you get your injection of this NP2 vector. These are patients with a terminal form of cancer, injected intradermally, so underneath the skin, into the dermatome associated with the pain. So at the location of their chronic pain. Dose cohort, so 10 to the 7th is the lowest, 10 to the 9th is the highest. So the lowest one has the highest pain ratings. Pain ratings are a subjective measure of how pain feels to people, but you can see the people who got a higher dose definitely are reporting less pain. And 10 to the 9th, that's a really high dose. It's really surprising that they could do that. Usually things are around 10 to the 3rd, 10 to the 4th for like a vaccine. But these are terminally ill patients, which means that if they choose to, they can take a riskier treatment, which I wouldn't call this risky because we know enough about herpes that there shouldn't be any control issues. But with new vaccines, there's always possibility of reversion. However, more standard vaccines like the measles vaccine, there's essentially no chance of that. This is a dose response curve, and so the higher the dose, basically, the better the treatment. So, what's wrong with it? Why isn't it used for everything? This latent infection isn't well understood. It seems like a perfect system for gene therapy because you can get the virus in, turn on latency, but get your transgene produced, but we don't actually understand it well enough to do that, so we can get the primary infection, get some of the transgene expressed, and then it might get shut down by this LAT gene. And then how do you target a specific form of neuron? So let's say you want to do, like I said before, the trigeminal neuron as opposed to a skeletal muscle neuron in your foot. And we don't really know. Amplicon vectors. So again, we talked about these plasmids before. They're mostly for gene therapy. And they're subject to the same transgene expression and targeting issues that we've already discussed. So they can be affected by the latent cycle and the neurotropic effect of the virus. Doesn't depend on the genome, it's based on the proteins on the capsid, which presents an interesting point that I don't know if we'll talk about in this little lecture. Advantages can potentially introduce up to 150 kilobases of foreign DNA, so quite a bit, and can insert really large genes with their endogenous processing signals. So you can bring in, normally when you put a gene in a vector, you only have the protein coding portion of the gene, but this allows for introns to be included, which means you can get splice variants and non-coding signals to be included. And along that line, you can include sequences that affect chromatin structure. So chromatin is the way that DNA is organized. Chromatin is DNA plus the proteins associated with it. And depending on how they're structured, it can limit access of polymerases to genes. And there's also potential for helper-free amplicons that could increase safety of vectors. So Normally you need a helper virus, so you need to infect with that replication defective version of HSV, but you sometimes will accidentally package one of their genomes with these transgenes, and so you'll actually be infecting your patient with that closer to wild-type version of the virus. And that would be bad, because you would cause an infection, and that's not your goal. And their largest limitation specific to the Amplicon vector is needing that helper. So let's just arbitrarily say orange represents the helper virus and yellow is just Amplicon vectors. And so how do you sort these? How do you separate them? Because these are identical, it's just the genome inside of them that's different. And if you can figure it out, you've got a nice paper waiting for you somewhere in the science community. So strategies for doing this is the defective helper virus I was talking about. Specifically, apparently, ICP27 is the gene you knock out. You grow stalks in a complementing cell line that's engineered to express ICP27, and then you can use Kralog, or you can use Kralog site-specific recombination to eliminate the packaging sequence of the helper virus. So you'll have the pack signal, it'll have LOXP site on either side, then you just add, you in fact create expressing cells. That way you can use the virus's wild type version of itself to grow up in a different cell type than before you're going to use it as this helpful helper virus. You can eliminate out the packaging signal and then infect with the next generation of virus into this cell line that you can put your Amplicon in. Or you can cofect Amplicon with viral DNA from a strain that already is a deleted pack signal. And so you start with helper-free, or you'll get helper-free Amplicon stocks. However, transfection is really, really limited in how much work it does. And so these titers is the amount of infectious virus produced, or in our case, the amount of Amplicon-containing virus produced will be really, really small. So, yeah, you can use multiple plasmids. I think that's just like a fancy name for multiple plasmids that each have part of the virus's expression of genes. So you can't accidentally make a virus because they're also plasmids. They're just expressing the wild-type viral genes and lack the plaque signal. So for the next generation of HSV1 vectors, you want retargeting, so you alter the target cells by changing viral envelope proteins and place HSV1 genes under control of a tumor or tissue-specific promoters. And people are working on that right now, and other viruses use that already. I know measles virus has had different envelope proteins put on it, and then this tumor-specific control is something everybody's racing towards. And so you want to have something like a gene that you know is only expressed in cancer, which acts as a transcription factor for HSV1 genes. HSV1 and AAV hybrid amplicons. So these amplicons can carry AAV rep site and the ITR sequences that it uses to replicate. And it allows for replication of the AAV1 site into chromosome 19. AAV1 does not have insertional mutagenesis because it targets a specific spot on chromosome 19. So this is an interesting idea where you could get the HSV1 type amplicon, which is much larger than what AAV can carry, and then AAV can integrate what it can carry into the host genome fairly safely. And then HSV with Epstein-Barr virus. Remember, these two are fairly well related in that they're both versions of herpes viruses, though they're in different subfamilies. Alpha for HSV1 and beta for EBV. So these amplicons carry the EB1 ABNO1 gene and their ORIP which Ev1 will cause RIP to go make the Amplicon go latent, allows for replication of the Amplicon in dividing cells, because only dividing cells will give it the signal to do so. Otherwise, this Ev1 or IP will always tell it to be latent. That's just the references that went into making this. Special thanks to Stephen Rice, who made this and gave this seminar. Alright, any questions? Feel free to email carriebradio@gmail.com, and I'll talk to you next time.